This first unit in biology is a bit of a mishmash of different topics that are just thrown together. One of those pieces of mishmash happens to be that of the microscope. We're going to learn some of the parts of the microscope here initially, but the idea being eventually that you would be able to actually use and focus the microscope as a practical part of any science or biology class. To get that process started, we're going to name a few of the parts because you can't really learn the process of focusing without knowing the names of these parts. And in doing so, we're going to start off with the eyepiece. The eyepiece is just that. It's what you're going to look into when you're observing something in the microscope. But importantly, you'll notice in parentheses here, we've got 10x. That simply refers to the fact that the whole point of a microscope is magnification, making things bigger and easier to see. And some, but certainly not all, of that magnification takes place there at the eyepiece. It's a magnifier. It increases the size of what you're looking at by a factor of 10. If we kind of work our way clockwise here, we've got some structural components here that are of little significance when it comes to usage. We've got the arm, and down here we've got the base. Not a whole lot to mention about those two pieces, but importantly we do have what is labeled as two knobs, but what appears to be one. So you're looking at this knob right here, and there is an outer portion to that knob, and then there is an inner portion to that knob. The outer portion is the coarse adjustment knob. And notice the way that the word coarse is spelled, C-O-A-R-S-E. Coarse in this spelling means rough. So the rough or coarse adjustment knob. You're going to use that knob in order to focus on some specimen, and you're going to do so in a rough sort of way, as in you're going to get the object that you're looking at into focus, but it's not going to be perfect. It's going to be a little bit rough. You will then use this inner knob, the fine adjustment knob, to fine-tune what you're looking at so that it's just right for your eyes. We've got the stage adjustment knob right here. That simply allows you to move the stage, which is this structure, in two separate planes, both right and left, and then forward and back, so that you can move the specimen that you're looking at without actually having to put your hands on it and mess up your focus. When seeing anything, you got to have light, so there's your source for that. And lastly, we have what's called the revolving nose piece, which is simply this structure right here that allows you to move different sized lenses into and out of position as you want to look at something under the scope. If we look a little closer at that revolving nose piece and the area around it, there are a few additional parts that you need to know. Let's start over here with the stage clip. That's this structure right here. That simply allows you to place the slide that you're going to be looking at in place, and it sort of holds it there and keeps it still. Let's skip down here to the diaphragm. The diaphragm would be, and I know it's difficult to see, but that little black lever that's sticking out from underneath the stage. The diaphragm allows for you to adjust the light that is penetrating through your specimen. Sometimes things look a little bit better with brighter light. Sometimes they look a little bit better with less light. So by moving that black lever this way or that, you can open or close the diaphragm, thus letting in either more or less light onto your specimen. This big black piece here is the stage, and all that leaves us with then would be the lenses that are attached to the revolving nose piece. You have one here with a little red circle around it, one that's slightly bigger that has a yellow circle, and then the biggest, which has a blue circle around it. Let's start with the smallest, that being the one with the red circle, and that is the scanning lens. The scanning lens, as is noted in the parentheses, magnifies the object that you're looking at 4 times. Now remember that we talked earlier about the fact that the eyepiece magnifies 10 times. So if you look through the eyepiece, which is magnifying 10 times, which is then looking through this scanning lens, which is magnifying 4 times, the total magnification is the product of those two numbers, as in multiply. 10 times 4 you're going to be viewing something 40 times bigger than it is with the naked eye. If we take one step up and look at this medium-sized lens, that is the low power lens. And you'll notice in parentheses it says 10x, which means if it's magnifying the object 10 times greater, and you're looking through the eyepiece, which is magnifying 10 times greater, you multiply those two numbers together, and the total magnification for that particular specimen would be 100x, or 100 times bigger than with the naked eye. Finally, we have the largest lens that we would use in biology, and that is the high power lens, with a magnification of 40x, of course multiplied by the eyepiece another 10 times, and therefore total magnification of 400x. Here are the steps that I would try to get you to follow in class in order to properly focus the microscope. Here are half of them. You will be seeing this in class and hopefully using them. And here are steps 6 through 10. We should be getting some practice with this by doing a lab. We're going to skip over some slides here, but they are merely directions for a lab that you will do in class, and we can skip those now. Having done that lab, some of the concepts that you would learn from doing that lab are listed here. Orientation. What I'm meaning by orientation when it comes to the microscope is that everything in a microscope is backwards and upside down. In other words, as the example here says, if you put the letter P under the microscope and looked at it, it would be flipped both upside down and backwards so that it would look like a D. Everything is in reverse. Another idea that goes along with the microscope is that of resolving power, simply defined as the ability to separate details. This is what your eyes do. You look at something and you see detail. The microscope, though, has a greater resolving power than your eyes do all by themselves. We can see details in the microscope that are too small to be seen with the naked eye. So it has a greater, the term might be resolution, or, as is stated here, resolving power. A couple of other ideas that go along with the microscope, one of which is depth of field. When you focus something under the microscope, you move the lens with that coarse adjustment knob and the fine adjustment knob. You're really moving the depth of that lens either closer or further away from the slide that you're looking at. Your lenses can only focus on one specific depth at any given time. It is hard to see much depth. This is why microscope slides are cut so thinly, whatever the sample happens to be that you're looking at. Your eyes actually do the same thing. If you want to focus on something in a microscope, you can focus on different depths. You can focus on something that's near the top of the slide. You can focus on something that's near the middle of the slide, but you can't focus on both of them at the same time. If you think about it, if you want to look at something without the microscope and you look at it real close to your eye, you can focus on it, but then things that are further away become blurry. If you want to pick your head up and look 50 feet away from you, you can focus on something that's 50 feet away, but then things that are very close to you become fuzzy. And so the microscope works in the same way. It's just a matter of magnitude. You move the lens a millimeter, and that makes a big difference when it comes to something as focused, as powerful as the microscope. Lastly, we have this idea of microscopes being parfocal. We mentioned earlier that there are different sized lenses and that we need to move them into a specific depth in order to get them in focus. The nice part about our microscopes being parfocal is that you can flip from the scanning lens to the low power and from the low power to the high power, and they should all be in focus at the exact same depth. When it comes to microscopy, there are different types of microscopes, and the ones that we've been describing here are light microscopes. Pretty simple idea. Light passes through a specimen, hits a lens, becomes magnified, and then that light hits your eye and you perceive it as something. Light microscopes are nice to be used for viewing living things, and they can also view things that are no longer living or never were, as in preserved specimens. Very simple idea would be a magnifying glass. What we use in this class is a compound light microscope. But light microscopes aren't the only type of microscopes. Here's an image from a light microscope. Here's another one of a cell dividing. But other types of microscopes besides light would be electron. And there are others too, but we're going to focus in on the electron. Obviously with a name difference, light microscopes use light. Electron microscopes do not. They magnify instead with electrons. The problem with electron microscopes is that they cannot view living organisms. They can view what were living organisms but are no longer living. There are a couple of types of electron microscopes, transmission being one, scanning being another. If we look quickly at a transmission electron microscope, you can read the definition here in that it sends a beam of electrons through the specimen. That, trust me, will be a key word. Because of that through, the image that you end up seeing from a transmission electron microscope is very two-dimensional, very flat. But the amazing thing about a transmission electron microscope is the level of magnification being up to 2 million x. You can see here an image from a transmission electron microscope, very two-dimensional, very 2D, very flat. Same thing here. Whereas a scanning electron microscope instead sends a beam of electrons across the surface of the specimen, sort of maps the surface, and thus gives you a more 3D instead of 2D image. Although it does that, it doesn't provide quite the magnification of the transmission. It can only magnify up to about 50,000x, but that's still of great significance. And you can see some of the images I have here of three-dimensional scanning electron microscope images, how there is some depth to them that you did not see in the transmission.